Welcome back. Let's continue. How do viruses replicate? Did you answer correctly? The major steps in viral replication are the same for all viruses. A single round of the viral replication cycle can be separated into several steps. These are attachment, absorption, penetration, uncoating, biosynthesis of viral components, assembly of new viral particles, release. The first step in viral infection is attachment, interaction of a virion with a specific receptor site on the surface of a cell. The virus must recognize an appropriate target cell, attach to the cell, and then penetrate the plasma membrane and be taken up by the cell. This table shows you some examples of viruses and their receptor on cells of the immune system. HIV binds to the CD4 molecules on immune cells. Rhinovirus, rhino means nose. The receptor is ICOM1, intercellular adhesion molecules 1. Epstein-Barr virus, the receptor is CD21 on B cells. Then is penetration. After binding, the viral particle is taken up inside the cell. This step is referred to as penetration or engulfment. Interactions between multiple viral attachment proteins, VAPs, and cellular receptors initiate the internalization of the virus into the cell. The mechanisms of penetration depends on the virion structure and cell type, which could include direct penetration, fusion, and endocytosis. The naked viruses bind to the receptors on the host cells and then directly penetrate the genome into the host cells. The enveloped viruses bind to the receptors and then fuse their membranes with cellular membranes to deliver the nucleocapsid or genome directly into the cytoplasm. Also, the viruses can enter the cell by receptor-mediated endocytosis. After binding to the receptor, the host cell can carry out the endocytosis and form an endosome. Then the viruses invade inside the host cell. Next step is uncoating. Uncoating occurs at the same time with or shortly after penetration. Uncoating is the physical separation of the viral nucleic acid from the outer structural components of the virion, such that it can function. Step 4, biosynthesis of the viral components. Once inside the cell, the genome must direct the synthesis of viral mRNA and protein and generate identical copies of itself. As what we have learned in the last session, there are 6 types of viruses. They are double-stranded DNA virus, single-stranded DNA virus, double-stranded RNA virus, positive sense single-stranded RNA virus, negative sense single-stranded RNA virus, and retrovirus. All the viral replication obey the central dogma, DNA to RNA to protein. The genetic information is transferred from parent to offspring via DNA duplication. Within the cell, genetic information is transferred from DNA to mRNA transcription and then to protein translation. Please note, reverse transcription could happen. Reverse transcription is the transfer of information from RNA to make new DNA. This occurs in the case of retroviruses, such as HIV. Most double-stranded DNA viruses use host machinery in the nucleus to make mRNA, which is translated by host cell ribosomes into proteins. Replication of viral DNA occurs by semi-conservative means. The single-stranded DNA is converted into double-stranded DNA and replicates like double-stranded DNA. Positive sense RNA resembles a mRNA to make proteins. One of the proteins is RNA polymerase. That makes a negative RNA template and then more positive RNA genome progeny and mRNAs. Negative sense RNA virion carries RNA polymerase. The viral RNA is transcribed into mRNAs and a full-length positive RNA template by the RNA polymerase. Double-stranded RNA acts like negative sense RNA. Retroviruses have positive sense RNA that is converted to complementary DNA, cDNA, by reverse transcriptase carried in the virion. The cDNA integrates into the host chromosome, and the host makes mRNAs, proteins, and full-length RNA genome copies. Newly synthesized viral genomes and capsid polypeptides assemble together to form progeny viruses. Enveloped viruses released by budding process. Virus-specific envelope glycoproteins are inserted into cellular membranes. Viral nuclear capsids then bud through the membrane at these modified sites, and in so doing, acquire an envelope. There are no special mechanisms for the release of non-enveloped viruses. The infected cells eventually lyse and release the virus particles by cell lysis. The number of the infectious viruses can also be evaluated with a count of the plaques produced by tenfold dilutions of sample, plaque forming units. The summary of the viral replication. Step 1. Virus attaches to a cell. VAPs bind to the receptors. Step 2 to 3. Virus penetrates cell membrane and injects nucleic acid, DNA or RNA into the cell. Step 4. Viral nucleic acid replicates using host cellular machinery. Step 5 to 6. New viral nucleic acids are packaged into viral particles and released from the cell. The host cell may be destroyed in the process. This is a short video about flu virus replication. Please watch the video and find out each step of replication. Can you figure out all steps? attachment, key and lock, right? Penetration, endocytosis. Uncoating, biosynthesis in the nucleus and in the cytoplasm. Maturation and release by budding. In this video, we can see just one virus. There will be millions of progeny viruses. But please note, not all infections lead to new progeny virus. One situation is abortive infection. That means to fail to produce infectious progeny, either because the cell may be non-permissive and unable to support the expression of all viral genes, or because the infecting virus may be defective, lacking some functional viral gene. Another one is defective virus. A defective virus is a virus that needs the help of another virus for the formation of the infectious particles. The most typical example is HDV. HDV cannot replicate in a host cell without the help of HBV. HDV is a defective virus. And interference. The interference is the effect that infection of either cell cultures or whole animals with two viruses often leads to an inhibition of multiplication of one of the viruses. Now, break time. See you soon.